Today we're going to talk about the development of bones. Bones begin to develop very early in the human embryo. Around the 6th or 7th week of embryonic development, a process called ossification begins. This is when the cartilaginous structures start to transform into bone. There are two main types of ossification, intramembranous and endochondral. Intramembranous ossification is responsible for forming the flat bones of the skull, whereas endochondral ossification forms the long bones of the body, like the femur and humerus. Intermembranous ossification is responsible for forming the flat bones, such as those of the skull and the clavicles. This process begins with mesenchymal cells, which are a type of stem cells. Mesenchymal cells group into clusters and differentiate into osteoblasts. These clusters become the ossification center. The osteoblasts secrete a substance called osteoid, which traps them, turning them into osteocytes. A trabecular or spongy bone matrix and a protective layer called periosteum forms. Eventually, compact bone develops on the surface of the trabecular bone, and the blood vessels within the bone condense into red bone marrow. Endochondral ossification, on the other hand, is the process that forms most of the bones in the body, particularly long bones like the femur, tibia, and humerus. This type of ossification begins with the cartilage model of the bone. The process starts at the primary ossification center, typically located in the middle of the cartilage model. This cartilage model serves as a blueprint or template for the future bone. It is a framework of hyaline cartilage that resembles the shape of the bone to be. Mesenchymal cells differentiate into chondrocytes, which produce a cartilage model of the future bone. Blood vessels grow around the edge of the cartilage model, bringing in osteoblasts that deposit a bony layer around the cartilage. Capillaries penetrate the cartilage, bringing osteoblasts that form bone within the cartilage model, creating the primary ossification center. As the bone grows, the cartilage and the chondrocytes continue to grow at the ends of the bone, while the medullary cavity in the center expands and remodels. Afterwards, secondary ossification centers develop at the end of the bone. Hyaline cartilage remains at the epiphyseal or growth plate and at the joint surfaces as articular cartilage. During childhood and adolescence, bones grow in length at the epiphyseal plates known as growth plates. These plates are bands of cartilage located between the primary and secondary ossification center. The cells in the growth plates continuously divide and secrete new cartilage, which is then replaced by bone, allowing the bones to lengthen. Once a person reaches adulthood, the growth plates ossify completely and the bones stop growing in length. However, bone remodeling continues throughout the life, ensuring bones remain strong and healthy. Even after the bones are fully developed, they are not static. Bones are living tissues that undergo constant remodeling. Old bone is broken down by cells called osteoclasts, and new bone is formed by osteoblasts. This process helps maintain the strength and integrity of our skeleton. Several factors significantly influence bone development and can impact skeletal health. Genetic factors are crucial. Mutations in genes like COL1A1 and COL1A2, which encode type 1 collagen, can lead to conditions such as osteogenesis imperfecta, where bones are brittle and prone to fractures due to abnormal collagen production. Nutrition plays a critical role. Adequate intake of vitamin D, calcium, phosphorus, and other nutrients is essential for proper bone growth and mineralization. Vitamin D, for example, is crucial for calcium absorption, necessary for bone strength and development. Environmental factors during pregnancy can affect bone development. Exposure to teratogens, medications, and toxins can interfere with skeletal development in the fetus. Maternal conditions like diabetes or maternal smoking can impair fetal bone growth and development. Mechanical factors such as physical activity and weight-bearing exercise stimulate bone growth and density. Lack of physical activity or prolonged immobilization can lead to decreased bone density and weaker bones. Clinically, disruptions in bone development can lead to a variety of conditions. Skeletal dysplasias, for example, are a group of conditions where bone grow abnormally in size, shape, or structure. Achondroplasia, the most common form of dwarfism, results from a mutation in the FGFR3 gene affecting the growth of long bones and leading to shorter limbs. Osteogenesis imperfecta, also known as the brittle bone disease, is caused by mutations affecting the production of type 1 collagen, the primary type of collagen in bones. This results in bones that are fragile and prone to fractures, often with varying degrees of severity. Another condition, called congenital pseudoarthrosis of tibia, is characterized by a non-union or abnormal union of the tibia bone. It is thought to be related to abnormal embryonic development of the tibia, leading to instability and frequent fractures.